Imagine, your phone lights up with a text message. It's from a trusted friend, and they're telling you to change your plans, drop whatever you're doing, and go to this specific address immediately.
Right, which you do because you trust them.
Exactly.
Yeah.
You trust the source, so you do it.
Yeah.
But what you don't realize is that, well, your friend's phone was stolen like hours ago.
Yeah.
The person texting you is a hacker, deliberately leading you right into a trap.
It's a classic phishing scam.
Yeah, and we experience this kind of, digital deception all the time, or at least we are warned about it constantly. But right now, at a microscopic level inside your own body, the exact same kind of hacking is taking place.
It really is. And it's a deeply unsettling reality when you start looking closely at cellular biology. I mean, we are talking about the ultimate breach of trust happening billions of times a second.
It's crazy. Welcome to the Deep Dive, everyone. Today we are unpacking this completely fascinating stack of research all about exosomes.
Which is such an exciting field right now.
It really is.
Yeah.
And for anyone who might just be, catching up on this topic, you can think of exosomes as the body's internal text messaging system. They are these incredibly tiny, nanometer-sized messenger bubbles that our cells constantly send to one another to provide biological updates.
Yeah, they're the communication network.
Right. But our specific mission today is to look at what happens when an active bacterial or fungal vaginal infection totally crashes this network. How does the presence of an infection just completely hijack this communication system? And why does this invisible warfare make rigorous safety protocols absolutely vital for the emerging field of exosome therapies?
Yeah, and to really understand the stakes here, we first need to strip away this old idea that exosomes are just passive cellular debris or, waste products.
Right, like the cell is just taking out the trash.
Exactly. For a long time, science sort of viewed them that way. But they are actually deliberate, highly engineered cargo carriers.
Okay.
They're packed with specific RNA sequences, functional proteins, and lipids.
They're very intentional.
Highly intentional. They are the primary way our cells coordinate really complex behaviors across different tissues. So when a bacterial or fungal infection takes hold, this communication network doesn't just get a little noisy, it becomes a literal battleground.
So let's start with the opening move of that battle, because before we can get into how the text messages themselves change, I noticed the research focuses really heavily on how an infection physically alters the neighborhood that the cells live in.
The environment, yes.
Yeah, the sources call it a metabolic hijack, and they look specifically at Candida albicans, which is the fungus responsible for a lot of vaginal infections. And from what I read, it seems like the fungus doesn't just attack the cells structurally, but actually goes after their food supply.
It is a devastatingly strategic attack. When Candida invades those vaginal epithelial cells, it aggressively depletes glucose in the local environment.
It just eats all the food.
Pretty much. It is intentionally starving the host cells of their primary fuel source. But, it goes much deeper than just stealing food. The fungus actively interferes with the internal machinery of the host cell by inhibiting key enzymes.
Oh, wow.
Yeah, two of the main targets are enzymes known as LDHA and PKM2.
Okay, wait, let's pause there so we don't get lost in the, the biochemistry alphabet soup. I know enzymes are basically the biological workers inside a cell, but what do those two specifically do? Like, why is the fungus targeting them?
Right, so think of LDHA and PKM2 as the critical foremen in the cell's energy factory. Their job is to manage a process called glycolysis, which is just how cells break down glucose for energy.
Okay, makes sense.
But crucially, in the vaginal environment, the natural byproduct of that specific energy-making process is lactic acid.
Lactic acid.
Yes. And lactic acid is absolutely essential. It is what gives the vaginal environment its natural protective acidity. That low pH creates a very harsh, completely hostile environment for foreign invaders.
So it's basically an acid shield. The body relies on that constant production of lactic acid to keep pathogens out.
Precisely. So now, look at the brilliance of the fungus. By shutting down those two specific enzyme foremen, the fungus halts glycolysis. The factory just stops running.
And so the shield drops.
The shield drops, lactic acid levels plummet, the local pH rises, and yeah, that protective acid shield is gone. This completely paves the way for the fungus to establish a much deeper infection and triggers massive tissue inflammation.
Man, so the fungus starves the host cell of its fuel specifically to shut down the factory that makes the body's natural defense.
It's incredibly clever.
That feels so calculated for mindless fungus. But How does this tie back to the text messages? Like, if the host cell is starving and the acid shield is down, what happens to the exosomes it normally sends out?
Well, this metabolic crash completely alters the biological state of those host cells. Because they are now in a state of severe metabolic distress, any exosomes they produce going forward are no longer routine maintenance messages.
They're not just saying hello anymore.
Right. The content of their cargo changes entirely. They become stress signaling vesicles. The host cells just start blasting out panic signals, which alters the broader immune response of the entire surrounding tissue.
Like firing off SOS flares.
Exactly. They are telling their neighbors, we are starving and we are under attack.
Okay, so if the host cells are screaming in panic, what are the hackers doing? Because the fungus is obviously smart enough to shut down the energy factory, but I saw in the notes that the pathogens are actually manufacturing their own microscopic mail.
They are. The pathogens are releasing their own extracellular vesicles, or EVs, and the sophistication of these pathogen exosomes is staggering.
Yeah.
Yeah. I mean, if we stay with Candida albicans for a moment, the research shows that the fungus produces its own EVs, but it dynamically changes the message depending on the density of the fungal population.
Wait, I read something about this that totally threw me. The fungus actually changes its behavior based on how crowded the environment gets. How does that even work?
It is entirely dose dependent. So at low concentrations of these fungal exosomes,
Like when the infection is just starting.
right, the research pinpoints it at around 15 micrograms per milliliter. At that level, the vesicles act as a growth signal. They promote the proliferation of the fungus.
Okay, so they're saying keep growing.
Essentially, yes, they are broadcasting a message to the surrounding fungal cells saying, the coast is clear, grow, multiply, expand.
But then when the infection gets worse and there are more of these exosomes floating around, the message flips, right?
It does. When the concentration gets high, which is around 60 micrograms per milliliter, the Candida EVs counterregulate.
Meaning they hit the brakes.
Yeah. They trigger the upregulation of a specific protein inside the fungus called NRG1. And NRG1 is a transcription repressor. You can think of it as a genetic brake pedal. When NRG1 is activated, it specifically suppresses the fungus's ability to develop hyphae.
Okay, hyphae are those long branching roots that fungi use to dig deep into tissue, right?
Exactly.
But why on earth would a pathogen send out a signal to tell itself to stop being so aggressive and stop digging?
Because if it doesn't hit the brakes, it risks destroying its own home.
Oh, wow.
Yeah, if the fungus continues to aggressively invade deep into the tissue, it can cross into the bloodstream, and that causes systemic candidemia, which is a lethal blood infection.
So it would kill the person.
Right. And if the fungus kills the host too quickly or, triggers an overwhelming fatal systemic immune response, the fungus dies along with the host.
That is wild.
By using its own exosome network to monitor its population density and hit the brakes on its own invasiveness, it ensures it can maintain a chronic, localized infection. It's managing its own growth to ensure long-term survival.
That is just, it's population control via microscopic text message.
It really is.
And it's not just fungi playing this game, right?
Right.
The sources also talk about parasites, specifically Trichomonas vaginalis, and their strategy seems even more direct. They don't just talk to themselves, they send exosomes that physically fuse with human host cells to actively manipulate our immune system.
Oh, the Trichomonas parasite is an absolute master of immune evasion. When its exosomes fuse with a human cell, they deliver this molecular payload that forces the human cell to alter its own chemical signals.
Like a hostile takeover.
Literally. Specifically, the parasite's exosomes induce the human cell to produce interleukin-6, or IL-6, which is a cytokine that suppresses acute inflammation.
Okay.
And simultaneously, they force the human cell to downregulate interleukin-8, or IL-8.
Let's unpack those interleukins for a second. What does turning off IL-8 actually do to the body's defense strategy? Like, what does that mean for you as the host?
Well, IL-8 is essentially a chemical flare. When tissue is damaged, cells release IL-8 to tell neutrophils, which are the body's first responder immune cells, to rush to the site of the infection.
Like calling the cops.
Exactly. So by downregulating IL-8, the parasite is cutting the communication lines to the police station. The body simply doesn't send the first responders because the chemical flare was never fired.
But wait, if the immune system is constantly patrolling, and scanning for foreign invaders, how do these parasite exosomes not get intercepted immediately? How are they sneaking past the body's security systems to deliver these payloads in the first place?
That's the terrifying part. Exosomes are wrapped in a lipid bilayer, which is a fatty membrane that looks remarkably similar to the membranes of our own cells.
Oh, so they're wearing camouflage.
Basically, yes.
Yeah.
To the patrolling immune cells, these vesicles often just look like normal cellular traffic. By the time the immune system realizes something is actually wrong, the vesicle has already fused with a host cell and dumped its manipulative cargo inside.
Man.
But the Trichomonas parasite does something else with its exosomes that is perhaps even more remarkable. It involves how the parasites communicate with weaker members of their own species.
Yes, the stickiness upgrade. I was reading about this. The research notes that there are different strains of this parasite, right? Like
Huh.
some are highly adherent, meaning they are really good at grabbing onto and sticking to human vaginal or prostate tissue.
Right.
And then some are poorly adherent. But the highly adherent ones can send out exosomes that get absorbed by the weaker parasites. And suddenly the weaker ones become sticky too. How does that biological cheat code actually transfer?
So we call this the horizontal transfer of virulence. In human terms, it's like peer-to-peer file sharing, but for biological
Oh
weapons.
my gosh.
The exosomes from the highly adherent strain are packed with the specific mRNA blueprints and sometimes the actual surface proteins that create that stickiness.
Wow.
When a weaker parasite absorbs that exosome, those lipid membranes fuse, and the new proteins are directly integrated into the weaker parasite's outer shell, or the mRNA actually instructs the weaker parasite to start manufacturing those sticky proteins itself.
They're literally passing out tactical gear to the rookies.
That is a very accurate way to visualize it, and this strategy is incredibly widespread. Bacteria do it as well.
Really?
Yeah, Gardnerella vaginalis, which is a primary bacteria involved in bacterial vaginosis, they use bacterial EVs to deliver concentrated toxins directly into host cells.
Oh, brutal.
Because the toxins are packaged inside the vesicle, they are protected from the local environment and are delivered with pinpoint accuracy, causing host cell death. The pathogens have entirely weaponized the exosome network.
So if we zoom out and look at the whole environment, it is completely flooded.
Yeah.
We have the stressed out human host cells screaming in panic, the fungi sending out population control signals, parasites sharing tactical gear, and bacteria lobbing toxin-filled grenades.
It's chaos.
It is a chaotic soup of biological data, which leads us to a consequence in the research that I found genuinely terrifying: the cargo exchange.
Yes. This is where the localized infection becomes a systemic threat because the pathogens are flooding the area with their own vesicles alongside, the physical debris of dead bacteria and ruptured cells. The environment is just full of biological trash.
Right.
And the host's own exosome network gets dangerously cross-contaminated by this debris.
Yeah, the sources mentioned that human extracellular vesicles can end up carrying bacterial DNA, or something called DAMPs. What exactly are DAMPs, and how does this trash get inside a human text message?
DAMPS stands for Damage-Associated Molecular Patterns. It is basically the molecular debris left over when a cell is destroyed or when a bacteria is broken apart.
Okay.
Now, human cells are constantly forming new exosomes by pinching off pieces of their own internal membranes. Sometimes this process accidentally scoops up the free-floating bacterial DNA or DAMPs present in that infected environment.
Just totally by accident.
Often by accident, yeah. Other times, the human immune cells intentionally package these foreign fragments as a way to present the threat to the rest of the immune system.
Like sending a mugshot.
Exactly. But either way, you end up with a perfectly normal, human-produced exosome that is now carrying enemy contraband inside it.
It's the stolen phone from the analogy at the beginning.
Uh,
The text message looks like it's from a friend, but the payload is hostile. So what happens when these contaminated human exosomes leave the vaginal environment and travel through the bloodstream? Like, if a distant cell receives this package, how does it react to the contraband inside?
This is where we see a profound breakdown of cellular trust. The distant, healthy cell sees a human exosome approaching. It recognizes the human surface markers, assumes it's a friendly maintenance message, and lets it fuse.
And then?
But the moment that bacterial DNA or DAMP is released inside the healthy receiving cell, it trips internal security alarms. Two of the most heavily studied alarm pathways are known as CGA sting and AIM.
Okay, let's translate those pathways. What do CGA, sting, and AIM2 actually do when they get tripped?
Think of them as internal tripwires designed specifically to detect DNA floating where it shouldn't be. In a healthy cell, DNA belongs securely inside the nucleus.
Right.
If these tripwires detect bacterial DNA floating in the main body of the cell, which is the cytoplasm, they immediately recognize that a breach has occurred. They set off a massive internal fire alarm, forcing the cell to release severe inflammatory cytokines.
So the distant cell is reacting as if it's under active bacterial attack, even though there's no actual bacteria there.
Exactly. This is a phenomenon called sterile inflammation. We call it sterile because if you were to swab that distant tissue and try to culture bacteria from it, you wouldn't find any living, replicating pathogens.
Because they're just not there.
Right. The bacteria never made it that far. But the genetic ghost of the bacteria was delivered via the exosome. This explains how a localized vaginal infection can cause systemic, whole-body inflammation or even impact a developing pregnancy. The human communication network has been totally compromised.
Man, if our own cellular communication network is this vulnerable to being compromised by a hidden infection, what does this mean for medicine? Like, we've been hearing so much about the booming field of regenerative exosome therapy.
Yes, it's huge right now.
Right, but if you're telling me that a hidden infection can turn exosomes into Trojan horses, how does anyone safely manufacture these therapies?
That is the exact dilemma facing the clinical field right now. The therapeutic potential of exosomes is undeniable, especially in regenerative gynecology. Treatments like exofemale are gaining major traction for vaginal rejuvenation.
Yeah, the sources mentioned exofemale. The goal of these therapies is to deliver targeted regenerative payloads that restore blood flow, radically reduce tissue inflammation, and help regulate that critical local pH environment we talked about earlier.
Exactly, rebuilding that acid shield.
And they use adipose-derived mesenchymal stem cell exosomes for this, meaning they're pulling these healing signals from fat tissue stem cells, right?
Correct.
It sounds like an incredible way to heal the body by just applying the healing instructions directly to the damaged tissue. But the extraction and isolation process seems like a complete minefield if an infection is present.
It is a profound logistical challenge. To isolate exosomes, laboratories have to separate them from all the other components in a biological fluid. But exosomes are incredibly small, typically between 30 and 120 nanometers.
Which is tiny.
Beyond tiny. Because they're so small, you can't just filter them out easily. Laboratories have to rely on their physical properties, primarily their size and their buoyant density.
Okay, and the two methods mentioned in the research are size exclusion chromatography and ultracentrifugation. How do those actually work in practice?
So, size exclusion chromatography, or SEC, is essentially a microscopic coin sorter. You pass the fluid through a column filled with porous beads, and it separates the particles based strictly on their physical size.
Okay.
Ultracentrifugation takes a different approach. It involves spinning the fluid at unimaginable speeds, often up to 100,000 times the force of gravity, to separate the particles based on their density.
But wait, if SEC is just sorting by size and ultracentrifugation is sorting by density, wouldn't a fungal exosome look absolutely identical to a human exosome to these machines? Like, we just spent this entire time discussing how pathogens make EVs that are virtually the exact same size and structure as human EVs.
You have identified the core danger. If you attempt to isolate exosomes from a biological fluid that contains an active or even a hidden infection, SEC and ultracentrifugation are virtually blind to the difference between a human exosome and a pathogen exosome.
They just can't tell them apart.
They can't. They will seamlessly co-isolate the pathogen EVs alongside the human EVs.
So if someone goes to an unregulated clinic that just, extracts their blood or fluid, spins it down, and injects the exosomes back into them for rejuvenation, and that person happens to have a hidden Candida or Gardnerella infection, they could literally be injecting concentrated pathogen signals into their own tissue.
They would be paying to be injected with an optimized biological bomb.
Oh my God.
Yeah, they would be delivering the stickiness cheat codes and the bacterial toxin grenades in a highly concentrated dose. This is a very real, very dangerous possibility in unregulated DIY exosome therapies. It completely defeats the purpose of regenerative medicine and instead creates a massive vector for severe inflammation.
That is terrifying. So how do the legitimate clinical-grade therapies avoid this? The research cites strict exosome care manuals, like the AMSBO guidelines. How do they ensure the servers haven't been compromised? To use the hacker analogy.
Well, legitimate therapies circumvent this risk by changing the source material entirely. Safe therapeutic exosomes are not extracted directly from potentially infected patient fluids.
Okay, good.
Instead, they are rigorously sourced from sterile, tightly controlled, lab-cultured environments. They use highly vetted human mesenchymal stem cells grown in bioreactors.
So they are building a closed-loop, sterile network from scratch rather than trying to filter a contaminated public network.
Precisely. And even then, the guidelines dictate extreme safety control procedures. The handlers must work behind protective screens in clean rooms.
It's very intense.
Very. And on top of that, every single batch of therapeutic exosomes must undergo exhaustive mandatory screening. They test for endotoxins, they require rigorous serological reports, and they perform broad-spectrum pathogen testing.
Just checking every single box.
Every box. The goal is to ensure absolute sterility before a single vial ever reaches a clinical setting. The regenerative power of exosomes to heal vaginal tissue is massive, but the purity of that signal is everything.
It really is. It just puts into perspective how delicate and complex our biology actually is. Let's take a breath and retrace this journey, because we started by looking at how an infection physically alters the environment.
Right, the metabolic hijack.
Yeah, a fungus like Candida shutting down the cell's energy factory to drop the body's acid shield. And then we saw how pathogens act as hackers, launching their own text messages to suppress immune responses and, literally share tactical upgrades with weaker parasites.
And then we explored the fallout of that invisible warfare, which was the cargo exchange.
Right.
We saw how human cells accidentally package bacterial debris, leading to distant tripwires being pulled, triggering sterile inflammation, all of which underscores exactly why the booming field of regenerative exosome therapy has to rely on uncompromising, sterile lab protocols.
Absolutely. If you're listening to this, I really hope it fundamentally changes how you think about your own body. Like the next time you hear the words microbiome or infection, remember that it's not just about germs sitting statically on your tissue.
Yeah, not at all.
What is actually happening inside you is a high-speed, invisible data war playing out at the nanometer level.
It is a dynamic, relentless exchange of information. And as in any war, whoever controls the communication network controls the territory.
Which leaves us with one final, incredibly provocative thought to mull over, because we've talked exclusively about the bad guys hacking the network today. But our sources noted something amazing about the good bacteria in a healthy vaginal microbiome.
The lactobacillus.
Yes, specifically lactobacillus. It produces its own exosomes, and research shows these good exosomes can naturally protect human tissue from viruses like HIV-1 by actively blocking their entry.
It is basically the body's own counterintelligence program.
Exactly. And we also know that scientists are now successfully engineering custom exosomes in sterile labs. So if we can build these vesicles from scratch, and we know how pathogens communicate, could the future of medicine be treating antibiotic-resistant infections by sending in our own custom-built hacker exosomes?
Oh, that's fascinating.
Right. Could we program a vesicle to speak a bacteria's own chemical language, infiltrate their network, and send a message that forces the pathogens to shut themselves down from the inside out?
Using their own horizontal transfer mechanisms against them?
Exactly.
Instead of a stickiness upgrade, we deliver a self-destruct sequence. It is a brilliant and honestly very plausible frontier for modern medicine.
The ultimate counterhack. A microscopic text message the bacteria definitely shouldn't open. Well, thank you for joining us on this deep dive, and we'll see you next time.